Good morning, everybody. So before I get started, today I will finish the nervous system. Chances are I will not get through all of the slides. I almost never do. So we will stop when I end today. In other words, you'll only be responsible for what I cover. Okay. So we'll just go as far as I can before I have to stop for the end of class. At that point, on Thursday and next Tuesday, I will discuss endocrinology, the second communication system. Then next Thursday, still a little bit away, we've got exam 1. All right, just to give you a warning. All right. So I've already posted homework 2. It'll be available at 10 a.m. this morning. I know that you're not going to look at it for at least almost a week anyway, but it's there when you get around to it. All right. So this is where I left off. We had discussed the action potentials, and we have focused on the graded potentials. And remember that the purpose of these potentials is communication, and there's 2 aspects. Graded potentials are solely concerned with conduction within a neuron. Okay. Then we have graded potentials, and they're critical in bringing information to the neuron. Remember your anatomy. So that means that the only place action potentials can occur is in an axon. They begin at the axon hillock, they end at the terminal bud of the axon. By contrast, graded potentials begin on the soma or the dendritic regions. They can get close to the axon hillock, but that's it. Right? And also, as I noted, they can be either depolarizing or hyperpolarizing, depending on which ion is regulated open. If it's potassium, it's going to depolarize. If it's going to be potassium, it's an example of a hyperpolarizing signal, right? The other thing is to remember that within these graded potentials, they can be summed, okay? The intent of summation is excitation of the axon hillock and conduction of a signal down the length of the axon, right? So what I want to talk about now is putting everything together, right? So here is a postsynaptic neuron. This is the big sucker here. Here's the axon. The axon hillock is right here, the beginning of the action potential capability. There are 3 innervating or afferent neurons, right? As an action potential comes down the length of those presynaptic neurons, when it gets to the terminal bud, there will be the release of neurotransmitter from each of these synapses, right? Now, not knowing anything else other than what we've been talking about for the last couple lectures, which one of these presynaptic neurons, if it fires, is most likely to initiate an action potential? Anybody? You got a 33.33 chance of getting it right. Yes. Pardon me? This one? This one?
Yeah.
Why?
It's closest.
Yeah, exactly. That's that simple. Okay, they're graded potentials, they're decremental. So they decay from their source. The closer it is, the less loss of signal. It's that simple. All right. Now, correspondingly, which is the least likely to do that, depending on whether you consider this one or this one furthest away, whichever one is furthest away. All right. Also remember summation. So remember frequency of action firings. So every time an action potential comes down the length, gets to this point, it's going to release neurotransmitter. Remember that those neurotransmitters are rapidly degraded by the enzymes that exist in the synaptic region, but if I increase the frequency, that means that those neurotransmitters are in that space longer. They're binding to their receptors and they are stimulating graded potentials. Okay? Now, the goal is to get to threshold potential here, say minus 55. versus the resting potential of minus 70 millivolts. So that means that if I have a single firing here, maybe there's enough, I'm generating enough signal to reach threshold, I get an axe potential. This one is too far away if it fires singularly. If I increase the frequency, I can create enough, I can sum enough signal that I might get here and work. Same thing for up here. Another way to think about it, summation works both ways. So summation, those are both excitatory postsynaptic signals. One about a postsynaptic signal, which is inhibitory, and then one that is stimulatory. Let's say that this one and this one both fire, correspondingly similar time frames. Okay, I have enough frequency with this one to get a threshold signal here to initiate an axe potential. This one fires at a similar time frame. As a result, there is no axe potential. Why? You're summing, so why? Remember, it's not just stimulatory, it's also can be inhibitory. So this is resulting in a depolarizing signal that would result in threshold potential here. However, this one fires and it doesn't happen. That's because this was an inhibitory signal. So that would be plus and a minus, and it now does not reach threshold. All right. So I'm showing this because this can facilitate. Those are the kinds of questions that you can expect next Thursday. Right. So I want you to become familiar with it. Okay. I'm not going to ask you to draw this stuff. I'm not going to ask you to give me a high level. I'm going to ask you to solve questions that suggest to me that you understand the material. All right. And these are examples of what I can ask. Right. Now, over the next couple of slides, they're just one-offs. This one-off is that, remember, there are two excitatory cells in the body, neurons and muscle cells. Right? We've been talking about neurons. We've been talking about neuron talking to neuron. You have an axe potential coming down a presynaptic neuron. It goes to the synapse. The postsynaptic cell is a neuron. We know how that works. We just spent 2 lectures talking about it. What about a muscle cell? The synapse looks exactly the same, whether that postsynaptic cell is a neuron or a muscle cell. So here's my question. It looks the same. It sounds like a duck. It looks like a duck. Is it a duck? Yeah. All right. This synaptic communication between a neuron and a muscle cell is identical to a neuron talking to a neuron. At the synapse, downstream, it's completely different. But you don't worry about that right now. When we get to the muscle section, we will talk about the differences. But in terms of neuron talking to a muscle cell, it's identical. There's no difference between a neuron talking to a neuron or a neuron talking to a muscle cell, right? Now, here is basically a summary that describes the differences or uniqueness between graded potentials and axe potentials. And let me go over this because I hold you responsible for it. You need to know, all right? So let's start with axe potentials. They're all or none. Once you get to threshold, they will go till they reach plus 30. They are discrete wavelets by definition. They're always what? They're always going toward 0. So they're always depolarizing. Always. And there is always a refractory period. Remember, we talked about absolute and relative refractory periods. They are triggered by voltage-gated channels that open. They are restricted to the axon. They begin at the axon hillock and they end at the end of the axon, right? Let's go over to graded potentials. Graded potentials, on the other hand, they have a variable magnitude, depends on how much energy is going in. They can be depolarizing or hyperpolarizing. They are going to be generated by like chemical gated channels or mechanical gated channels, etc. they're not self-perpetuating like action potentials because of those, remember those relays of voltage-gated channels, they're decremental. That means they decay from their point of initiation. And basically, this means that they do not, I repeat, they do not have a refractory period, and therefore they can be summed. All right. So these are the basic differences, and I expect you to know them. Okay. The nervous system is, think about it as just neuropathways. We've talked about circuits. So these are some examples of those circuits. Those of you who have an ECE background, these look very familiar to you. This is just in the case of neuropathways. So let me kind of walk through them very briefly. So we can have initial input site that then rapidly diverges but stays in the same pathway. B, we can have divergence from a single initiating cell source, but which goes in different multiple pathways. We can also have convergence from multiple sources onto a single output cell. We can have convergence from a single source. Remember that these axons can branch. Instead of converging on multiple efferent cells, they can converge again, even though they're splitting up, they converge back again on the same output cell. Remember, it's just a matter of the amount of, you have a fixed amount of neurotransmitter released by every action potential. So the more of these synapses you have, that means you're going to have more neurotransmitter. You're going to generate a stronger gradient potential. Now we also have what's referred to as a reverberating circuit. This is responsible for making a self-perpetuating signal. So we initiate the signal, and part of the output is to come back and re-stimulate that to keep this pathway alive. Lastly, we have parallel after discharge. So all of these are representative of the various neural pathways that exist in your body. Right? Now, let's think about this in terms of pathways. So most of the time I've talked about the individual cell. I've given you an inkling, but let's talk about this. We have a cell. We talk about it being an afferent or an efferent cell. Is it presynaptic? Is it postsynaptic? It all depends on where you're at in that pathway. So let's take, for example, this cell here. It is a postsynaptic cell for those axons that terminate on it. Notice the colors. There's at least 4 or 5 of those. Each different color represents a different presynaptic cell. Right? Each of them can, when it fires an action potential, the result can be a release of a neurotransmitter. Now, one neuron is either going to result in a postsynaptic inhibitory or postsynaptic excitatory signal. It can't be both. It has to be one or the other. All right. Now, if we reach threshold potential axon hillock, we get an action potential that goes down the length of the axon to the end of this. This is the postsynaptic neuron, but it is the presynaptic neuron for every cell downstream. So neurons, depending on where they're at that pathway, can be both a postsynaptic as well as a presynaptic. It all depends on where they're at in that pathway. Now, we've talked about a simple classification of neurons from afferent in the outside of the central nervous system. We've talked about intraneurons, which are fully constrained within the central nervous system, and then we have the motor neurons. All right, remember that the efferent and afferent neurons are part of the peripheral nervous system. However, some aspect of them has to exist within the central nervous system since they are communicating to or out of the central nervous system. They have to communicate with the interneuron, which is fully restricted to the central nervous system, right? So that gets us to the subject of a reflex arc. All of you have exhibited this. You go to a physician's office for your annual checkup. They put you on the bench. They're talking to you. And next thing you know, you feel a little pressure on your knee and your knee goes up, right? That represents a reflex. Why do they do that? To see if your reflexes are normal. All right. How does it work? All right. So this is pretty simple. It's a very simple context whereby basically, if you note up here, in terms of the knee, it's the simplest circuit. You have an afferent, which means there's an afferent neuron that starts in my knee, goes up, and then it goes in the central nervous system here. Then it communicates directly with a motor neuron that goes out, that induces contraction of the leg muscles in my thigh, and I kick up. All right. That's a very simple arc. What's important to realize is these reflexes are independent of voluntary control. You did not know that little rubber hammer hit your knee because you weren't looking at your knee. And your reflex was automatic. All right. Now, the same thing can happen up here. And basically in this particular case, it's more of a standard. We have 3 levels of communication. We have the afferent, the interneuron, and the motor. All right. Now, it gets a little bit more complex. And it gets a little more complex in this regard. You're cooking dinner tonight. You're not paying attention. You touch your finger to the hot element of the stove. All right. You're not paying attention because you're looking off in some other direction. However, what happens is your hand jerks away. And only secondarily do you realize it's painful. And this is now an arc. Now, let's talk about the complexity involved in this reflex arc, right? So your hand is here. You need to lift it up. What's involved? First off, here is the humerus bone, radius the ulna, elbow joint, and we'll restrict ourselves to 2 muscles, the biceps and the triceps, all right? So if I'm holding my arm out like this, in order for me to extend or bend my forearm up, I have to first contract this muscle. In order for me to lift this arm, I have to generate enough contractile force in order to overcome the load resistance of my forearm. Because the bicep starts here, is connected down here, it bridges across this elbow joint. If I overcome load resistance, I pick it up. I can pick up that load and I bend my arm. What else is also involved? There is not only the contraction of this muscle. What about this muscle back here? If both muscles contract at the same time, what will happen? They generate the same contractile force. If they both contract at the same time, they both equally are applying pressure. That means that even though I'm trying to bend this way, this is bending this way. So there's no net movement. So what has to happen, getting more complex, when this one relaxes, in order for me to overcome load resistance, I also have to overcome the load resistance here. So what happens is I need to promote contraction here while promoting relaxation here. So this muscle will stretch. And then I can actually do this. So when you have this arc, it's a little bit more complex. Not only is it sending an afferent motor neuron, which is going to induce the contraction of the biceps, it's also simultaneously sending a signal to the triceps muscle for it to relax. And because of this dual interaction, we can move our arm. All right. Again, another one-off blood-brain barrier. I introduced this when I talked about glial cells. I said astrocytes are responsible for helping to make or create this blood-brain barrier. First off, why do we need it? All right. Your brain is highly protected from the rest of your circulatory system. Why? Because of parasites and other invading organisms, you want to restrict things that could get there from the blood. Because if your brain gets compromised, you get compromised. So you have a blood-brain barrier. That means that the capillaries in your central nervous system are unique to those outside. So if I have capillaries here in my skeletal muscles, the capillaries actually have these pores, which allow for things to go through those pores as well as through the cells. And we've talked about transport through cells. So basically, this is a tissue outside of the brain. In the brain, it's quite different. We need to protect the brain, so we now first have not any open, but we have tight junctions between neighboring endothelial cells, which forms a pore-free blood vessel. But in addition to the blood vessel itself, there is surrounding that these processes from the astrocytes. This creates what's called the blood-brain barrier. This means that only certain things can cross from the bloodstream and get into the brain. Right? Now, from a protective standpoint, that's great. We evolved that way. It's very protective. But if you, as a biomedical engineer, are interested in drug delivery, let's say, of proteins to the central nervous system, that's a challenge you have to overcome. Because if you can't cross from the blood into the cerebral spinal fluid into the brain, what do you have to do? Drill a hole in the skull and inject it. And I don't think your patients are going to want to have that done. So you have to come with different strategies to overcome this barrier. Now, nature has solved it. Remember, I introduced extracellular vesicles. Nature evolved them in order to transport the cell, even though they're large at, say, 100 and 150 nanometers, which is huge, they still can cross across here. Nature's designed them to do that, all right? When there are different researchers, different companies that are trying to utilize this approach in order to deliver drugs to the central nervous system, all right? Now, next one up. Can the nervous system repair itself? Can the nervous system repair itself following injury? And the short answer is yes, with limitations. So let's start with the peripheral nervous system. We have neurons going from here to here. Or let's say for here. I cut my finger. I cut across a neuron. What happens? It all depends on what side of the neuron is the soma. Let's say the soma is over here. That means that everything on the post-cut side over here, away from the soma, it doesn't have all that cell support. It's going to die. That axon is going to die. However, remember, there's a myelin sheath that surrounds that axon. Those are independent cells. They survive. They're retained. They actually then provide guidance because there's a cut here. When the cut heals, this axon has to then re-extend itself back all the way up here. It needs a guidance, and that's what those Schwann cells in that myelin sheath, it forms a tube structure which helps to guide for that. Now, the process takes a lot of time. It takes a lot. You got a long way to go. It takes a lot of time. Now, I like to pose this question. I've got two individuals. The first one is using a chainsaw out in the woods, cutting firewood. He has an accident, cuts off his forearm, his left forearm, right here. Okay? He has the presence of mind to put a tourniquet on, and he throws his hand into his ice chest because that's where his lunch. He goes over here to UPMC. UPMC has a surgical group which then will reattach that hand. Okay? Remember, it's his hand. There's a second individual that is due for a hand transplant. That means they, again, go to UPMC because UPMC does hand transplant, not as much as they used to, but nevertheless, they do hand transplants, which means that there are two surgical groups. One of them is going to harvest the hand from the donor. This is the dead body. And it's a surgical team, multidisciplinary, because you need an orthopedic surgeon, plastic surgeon, blah, blah, blah. All these people are removing this hand. Then it's turned over to a second group, which attaches it to the recipient. Okay. Now, my question is this: of those two individuals, who do you think has a better functional outcome compared to the pre-injury or pre-surgery? Anybody? 50-50 chance.
This one,
For why?
because the system stays the same for the people lose the hand and do it.
It's his hand.
Yes.
It's his hand. From an immune standpoint, you're absolutely correct. But from a functional standpoint, you're not. The reason being is this. Yes, initially, the person who gets his hand reattached, it's his own hand. So we don't have to give immunosuppressive therapies. Okay. The transplant you do. However, it all depends upon the quality of that hand. Remember, the person who was using that out in the woods, it got lopped off, it landed out on the dirt, and it took a few minutes before he threw it on the ice. It took him an hour to get to UPMC. There was ischemic downtime. That hand had significant damage. Plus, have you ever seen what a chainsaw will do to a piece of wood? Imagine what that will do to flesh. So it tears stuff up. So there's a lot they had to really cut back to get through the torn, damaged, traumatized tissue. Okay. Now, plus they had to figure out which vein goes where, which nerves go where, whatever. Okay, we get to the transplant. The group that is disconnecting that one hand from as they disconnect it, they're labeling the nerves and the vessels and so forth. And then as the other group is removing the hand from the recipient, are doing the same thing. So now they just connect A to A, B to B, C to C, and it's under sterile conditions, ideal conditions. So that's the better outcome. So what are the outcome expectations? I said it takes a long time. The expectations are this. Even the better outcome will be with a hand transplant, but 2 years out, ideal outcome, present technology means that you will have achieved maybe 30% recovery of functionality. 30%. That's with extensive OT, PT, training, and because number one, it just takes long. Because remember, everything downstream is going to die, and it's going to have to reinnovate from the host going back down here. And that takes time. Whether it's the autologous or allogenic, it doesn't matter. Okay. Central nervous system. At one point, where I was sitting where you were many decades ago, we thought that when you lost a brain cell, that was it. We didn't know about the concept of stem cells in the central nervous system. Those were the last ones to be discovered. That was back around the early 2000s. We now know that there are stem cells residing in the central nervous system. They have the capacity to facilitate repair, but not even to the degree that we can achieve with peripheral nervous system repair. So getting them to work appropriately, there's still big issues. But there is the potential somewhere we're in the far future where we can potentially have some regeneration within the central nervous system. Okay? Let's move to a different subject. Let's talk about sensing. Okay, so remember, one of the roles is to bring information into the system. So let's talk about receptors and I'll just be a little bit, and then I'm going to focus on one specific subtype. We'll talk about eyesight. But now let's do some general discussions first. Okay, let's talk about sensory receptors. Now, when we talk about that, we talk about functionality of those receptors. Now, they have to be transduction. Now, this means that they're bringing information in. That means it has to be graded responses. That means that we have to have some way of varying magnitude, signal strength. Number two, we have to be able to amplify that signal, right? We have to amplify that signal such that we can initiate an action potential. We then have to transmit that signal along the length. And then, of course, we need to go from outside, because remember, it's in the peripheral nervous system. We have to get it to the central nervous system. And lastly, we need to integrate that with some sort of response, okay, which ultimately means it's got to come out as a graded potential somewhere downstream. Okay, we can classify receptors based on what is the stimulus, or rather the modality. So we have chemoreceptors, which we've already talked about. We have thermoreceptors, which is, of course, temperature changes, nocioreceptors, which is pain, and then we have mechanicoreceptors and photoreceptors. Mechanicoreceptors are helping you to set up in those chairs. And then, of course, photoreceptors are restricted to your eyes. Now, what is the origin of the stimuli is next. Is this stimuli within your body? This means that they are enteroreceptors. Is it responsible for you maintaining posture and so forth? Those are proprioceptors. If it's coming from the outside, those are basically exteroreceptors. All right. And then, of course, there's distribution. Are they generally distributed throughout the body, or are they then the special senses? And the special senses are restricted to the head for the most part: taste, eye, hearing, etc. Now, what's involved in perception? Perception means that basically you're going to perceive some signal, right? So first off, you have to have detection. So you have to be generating enough stimulation to initiate an action potential. They have to sum those components. Then we have to be able to have magnitude of estimation. So that's signal strength in terms of action potentials, that's frequency. Then we have to have this frequency coding. So basically, so as we increase the stimulus, we can increase the output. We need spatial discrimination. Where is this coming from? And then we have to do feature determination and then feature abstraction. All right. I used to do a fair amount of robotic vision back about 10, 15 years ago. And basically all of this is stuff that your eye and your brain does. You never think about it. But translating that to a robotic system where it doesn't know anything, you have to understand each of these things. They become critical, important parts. All right. In addition to what I just talked about, there is quality of discrimination, pattern recognition, and sensory adaptation. So, for instance, pattern recognition, facial recognition. The earliest facial recognition software came from here. All right. One of my old work partners was Takeo Kanade. He was the father of facial recognition. They used to show stuff where there'd be a football, and they would then identify people's faces and so forth like that. I mean, it was fabulous. It's gone so far now, but this was like 15, 20 years ago. So basically, it all has to do with pattern. Think about it. If I look at each of your faces, I have the ability to process that and say, you are a unique individual. Then I have some memory of that, and then I recognize who you are as being a unique, different person. This is not easy to train a machine to do. It has to do all of those things I talked about in order to result in a pattern recognition. Okay. Now, what is sensory adaptation? So that's the way your body tends to protect itself. Give you a perfect example. You lost your apartment, and you have to go find a new one. All right. You end up getting apartments on the busway. It's on the city busway. And the buses run 24/7. So you try to go to sleep at night, you get down, just about the time you get to sleep, here comes a bus coming by. And for the first week or so, you are miserable. You just can't rest. You can't sleep because of all this noise. All right. About 3 months later, you have someone comes to visit you and they're bitching about all the noise. You say, what noise? I slept the whole night through. This is sensory adaptation. So what happens is because you're hearing, you're getting the same input day after day after day, your body, your mind, your brain says, okay, this is non-critical information. Let's just ignore it. Okay? And that is your sensory adaptation. Right? Okay. So what do these receptors kind of look like? Don't worry, we're not going to go in a lot of detail here. I'm not going to ask you specifically this on an exam. So you kind of just relax and listen for just a moment. You don't have to remember so much. So basically, we have general senses, which can be touch and pain and so forth. They can be unencapsulated nerve endings. That means you have exposed neurons, right? So these are usually for the general senses, and then they can be hair, your hair follicles, etc. Then on the other hand, we can have encapsulated ones, and basically that means you have these dendrites. They are wrapped with glial cells and other connective tissues, and they form a protection, and they have specific functions. Don't worry about the details. All you need to do is take home is you have the unencapsulated and encapsulated receptors. And those are nerve endings, right? Now, wait a minute. An actual receptor, we're generally talking about this form here because here we have a receptor here. When this is activated, we would initiate an action potential. It goes down the axon. However, you can also have a receptor, which is an independent neuron. Here over here, we just have this sphere here indicates a receptor cell. When it is activated, it does not initiate directly an action potential firing. It will release its neurotransmitters. That goes over here and initiates an action potential potentially on this secondary cell here. So there's two ways we can have receptors. We can have receptors that are part of the primary afferent neuron, or they can be independent receptor cells. Now, when we talk about signal discrimination, what we talk about are receptor fields. So these are the areas which are innervated by a single or a small group of afferent neurons, right? Now, these receptor fields are important because, for instance, you have a more dense receptor fields where you need to discriminate between when you want to have more sensitivity. Okay? I used to do this years past. I don't do it anymore. So I would have a volunteer come up. You know the double pointed compasses. You got pins on both sides, right? Not pencil, but pins on both sides. And you have a ruler. And if you start over here, you have the person there blindfolded, they can't see. You take the points, here are the 2 points of the compass. If you point them here, you can feel 2 points of contact, right? Measure that contact. Gradually get smaller and smaller. At what point can you no longer distinguish 2 independent points of contact? At your fingertip, it's pretty bloody small. It's on the order of a millimeter. All right. Now, let's move up here to my forearm. My forearm, if I start here, 2 points of contact, 2 points of contact, I get a bit, and I get down to about 2 or 3 centimeters. All right. Let's go to my back. Same deal. However, you find as I move to the back, it's much, I can only discriminate about this far, my forearm this far, my fingertips this far. Now, how does that impact physiological function? What is the purpose of your hands and your fingers other than grasping? Sensing. It's designed for sensing. And that's why you have these huge receptor fields. You don't go around sensing with your back. All right? Now, I want to talk about a concept called lateral inhibition. This is further discrimination of fields. So you have these receptor fields, and you want to say that within this receptor field, I want to say, well, if I press here, how do I know I'm pressing at this part of the field or the whole thing? How does your body then narrow that down? Okay, so remember graded potentials. Graded potentials depends on the firing frequency of action potentials. So I need to stimulate receptor fields in order to generate those action potentials. The more firings of those action potentials means that it's more likely to continue communicating in that direction. So in lateral inhibition, what happens is here's a receptor field. And even though I'm pressing here, remember the graded potential, the most input is at the point directly underneath the pin. And it's decremental away from that. So we get further away, less strength of signal. Now, the way lateral inhibition works is the greatest frequency of action potentials is at this point. So it's going to go here, it's going to keep going. Now, lateral inhibition means that there's a slight stimulation here and here, but the frequency is not near as good here. Now, what happens is this, as we increase the frequency, that means that in this particular case, we have a stimulatory, but when it comes across here, we'll actually act to block these on neighboring aspects. So what that essentially does is narrows the stimulation to a single point. That's lateral inhibition. Now, this is a hard concept, so I've given you a couple of additional slides so that it'll help you do it. So all you really need to understand about lateral inhibition is that it's a process whereby the point at which you give maximal stimulation, get maximal frequency of action potential firings, that's going to direct it down that single pathway and provide for lateral inhibition at the less sensed pathways, right? Now, sensing, one of the things we sense is pain. So let's talk about pain. What is pain? We sense pain with receptors which are called nociceptors, right? They are throughout your body with one exception. You do not have nociceptors in your brain. Okay? Now, these obviously tend to be fairly rapid in signal conduction. And basically, we have fast pain, we have slow pain. We don't necessarily, of course, from my fingertips, I want to have very rapid communication. In my gastrointestinal tract, I don't need speed. Speed is not important, so things can be slower. So somatic pain is like in our muscles, in our joints, and then we have visceral pain, which is in our gut. And then, so basically, this is pain, all right? So, in terms of visceral pain, like say in my abdominal cavity, there are receptors there. However, when, for instance, if you're cutting on the stomach or something like that, the pain is referred. Okay, so that and the visceral pain is referred to as somatic aspects as a point of reference. Okay, these are called pain maps. They've been around for decades. All right, so basically what happens is this. So when you have a pain in the colon, and the colon is way down here, it will be about mid-abdominal. And then appendix, it can be, it will be a little bit over here, and so on. So, for instance, the stomach can be, for instance, if you have heartburn, it has nothing to do with your heart. Because what happened is the regurgitation of acidified contents from the stomach into the esophagus, and that's painful right here, but your heart is right here, so you call it heartburn just simply because of orientation, but it has nothing to do with the heart, right? Now, the thing about it, these are generalized reference maps. For instance, let's say your gallbladder is up here. So usually the pain is here. But it doesn't have, not everyone expresses gallbladder pain here. Sometimes they're off quite a bit. Now, depending on how your physician notes the pain, they will say, oh, your pain was in the right place. But you have to remember, those are generalized maps, and everyone here may not act the same. All right? So it's only an approximation, and I've known of more than 2 or 3 instances where people were misdiagnosed because the physician misinterpreted the pain. All right? Now, headaches. Well, wait a minute. I have a headache, but I don't have basically pain receptors in my brain. because the headache you're having has nothing to do with brain injury. So I used to have better than 20/20 vision. I use my eyes I use for a living because I'm reading papers, et cetera, et cetera. In my mid-30s, I would come home at the end of the day and I'd have a splitting headache. A splitting headache. I had no idea what was going on every day. Of course, on the weekends when I wasn't reading as much, I didn't get a headache. Then I come back on Monday, come back Monday night, I have a splitting headache. So I just happened to have to go, had an ophthalmologist friend. He said, come here and get in the chair. He analyzed and said, you need reading glasses. Like, okay. And that's because your eye strain. The eye strain, we'll talk about the eyes in a moment, but the strain was such that because I am overstressing my eyes in order to force them to see close up, it resulted in pain. All right. Headaches we're heading into flu season, cold season. A lot of times you're going to get congestion up here in your sinuses. That's going to result in swelling of tissues, and that's going to be pain. And that can be headaches as well, right? So even migraines and so on, but these do not have to do with actual brain pain sensation, right? Now, I want to talk about the senses. Now, we have a lot of senses. I'll mention taste. I won't touch on hearing. It's not that it isn't important. It's just that I only have a limited amount of space, time frame. So I'm going to talk about eyesight. All right. Now, I want to talk about vision. Now, vision and light. You're seeing light rays. And basically, what this means is vision is detectable within electromagnetic radiation. You're sensing electromagnetic radiation between 400 and 750 nanometers, right? Now, how do I perceive light? It's because these electromagnetic rays will stimulate photoreceptors in the eye, right? And this then results in the creation of a neurosignal that goes to the brain. It gets processed, and that allows us to see you in front of you, and you see me down here. Now, why 400 to 750? Not every animal sees in this range. We're human. This is our visual light range. Anything below 400, that has so much energy, it's going to kill those photoreceptor cells. Anything above 750 is not enough energy to excite those photoreceptor cells. So let's go back to 5th grade physics. Okay. Let's talk about basically light rays. Let's talk about convex and concave lenses. All right, because we have to talk about that to understand eyesight, how your eye works. So all of you are far enough away. By the time the image, the light rays, the image that represents you gets to my eyeball, all those light rays are parallel. Okay. They go through the lens of my eye, which is up here. It is this convex lens. Right? How does a convex lens work? What it does is we have parallel light rays coming in. As they pass through the convex lens, they get focus coming out. So they become convergent light rays. Ultimately, they become focused at some point. And that is going to be on the back of my eyeball. And when those converge, that allows me to see something clear, in clarity. Right? So that's basically this convex lenses. All of you, have you ever used those microscope, what do you call those lenses that, with the rain you hold it, you made fire with them as kids? Okay. So basically those are convex lenses. So I used to get great glee out of starting fires with those when I was a kid. You hold in the sunlight, you hold them appropriate, if you go down low, or put it on your hand. If you go down low, it's unfocused. it doesn't feel warm. As it's getting closer to being focused, it feels hotter. And at one point, you will actually can generate enough heat to burn your hand. You can start a fire with it. All right. So basically, that your eye is a convex lens. It is designed by nature to receive parallel light rays, things far away. Okay. Now, what happens if things are close? That means they haven't achieved parallelism yet. That means they're divergent pathways. If those divergent pathways are going through this convex lens, they will converge, but because they're not parallel, that means their point of convergence is further back than if they were parallel, which simply translates to they see a blurry image. Right? So, well, wait a minute. How do I adjust that lens such that I refocus those divergent light rays to still end on this particular focal point? I need to increase the strength of that lens. So I increase the angle of convergence so that it now then meets, even though they were divergent coming into the eye and not parallel. All right? Now, I've talked about convex. What about concave? In concave, we have the inward aspects instead of an outward angle of it, and that results in creating a more divergent set of light rays. Okay? So that means that instead of having convergent light rays coming out of it, you will have divergent pathway light rays coming out. So what's the difference between, I want you to know what convex and concave are. The easiest way to remember the difference, concave means in. So they're in, so that's concave, and convex is out. Right? So with convex, it will create divergent light rays when the light passes through. Concave, you will create divergent pathways when light passes through. And these will play a critical role as I move to how your eye works. Okay? Now, you have your eyes. Each eyeball has a lens. That lens has the capability of seeing images far away and close up. So that means it has the ability to see both parallel light rays as well as divergent light rays. As I just said in the prior slide, the only way I can adjust the focus of those divergent pathways is to increase my lens strength. You have a single lens in your eye, yet it can see things close and far away, which means it can change its strength. And this is how your eye can see things close up as well as far away. The default eyesight for humans is things far away, right? So your lens is, its default setting is to see things far away. These parallel light rays come through this convex lens. They're focused on the back of the eye. At the retina, you see a clear, distinct, crisp image. All right? If you move up close, we have to increase the strength of the lens. And the lens of the eye self-adjusts in order to see that. So it increases the it increases the strength of the lens. This process whereby it can go from parallel to divergent capabilities is referred to as accommodation. Now, this is enabled because of the unique anatomy of the eyeball. So here is your lens. It's in the middle of your eye right here. And it is suspended in place. So there are these suspensory ligaments that are bound to the lens, and then those are bound to what's called the ciliary muscle. The ciliary muscle is one of the only circular muscles in the body. So I've talked about the biceps here. It has a beginning, it has an end. With a circle, think of a hula hoop or a rubber band. There is no beginning, there is no end. It's a circle. All right? Now, When your bicep contracts, it physically shortens in length. What happens to a circle when it contracts? The entire length does change, but the length translates to circumference. So the circumference is being reduced. As the circumference is being reduced, what's happened to the diameter? It's being reduced. Now envision, we have our lens. It is being stretched by the ciliary muscle, by the interacting ligaments here. So it's being stretched, right? In the process of accommodation, what happens is I will induce the contraction of this ciliary muscle. That means it gets smaller. That means there's less tensile force on those suspensory ligaments, less tension on the lens, and it will then get fatter because it's not being stretched, because it's a big hydrogel. So that increases effectively the strength of the lens, right? Bless you. So you can see things by default far away without changing the strength of your eyeball. But as you get to close to you and you're seeing divergent light rays, you have to increase the length, the strength of your lens, and thereby through the process of accommodation, we reduce the diameter of the ciliary muscle, we reduce the stretching force on the lens, and it then becomes thicker, stronger, and we can see both parallel and then close or divergent light rays. All right. So this is critically, you need to understand this process, right? Okay. Let's talk about then eyesight. And when things, a good number of us here have some corrective lenses, whether it's glasses or contact lenses. Some of us have dual stuff going on as well. You get old enough, it'll happen to you too. Okay, so let's talk about that. All right. So normal eyesight, things far away, you have parallel light rays. Your eyeball, your default strength of your eyeball is fine. Light rays go and what happens is they then converge and they then are focused on the back of your eyeball and you see things far away clearly. Okay. Now we get up close. We have divergent pathways. Now the eye accommodates. There's contraction of the ciliary muscle that results in an increase in lens strength. That increases the angle whereby you can now see. All right. So thereby you can, within the eyeball, you can see things both far away and near. Right. So let's talk about two specific categories, the most broadest categories. So let's talk about nearsightedness, which is also referred to as myopia. Right. And we'll talk about that first. In nearsightedness, in the uncorrected, there is something wrong with the eyeball. All right. So basically, that means that you can basically, the thing about it is that your eyeball lens its default strength is too strong, or the eyeball physically it's kind of an ellipsoid and it's too long, right? What does it mean? Let's just take if we have an increased strength, that means that if I have my light rays coming in there parallel and I have it too strong, that means that the focal point will be ahead of where it's supposed to be. Or if it's too long, again, the focal point is not on the back of the eyeball, on the retina. It's ahead of that, which means I'm going to see a blurry image. All right. So I can't see things far away. I am nearsightedness, but that means that, well, wait a minute. I can't see things far away because my lens is too strong. But now I get up close, that strong lens is enough to see things close up. So I need to accommodate because my lens is already strong enough. But I can't see things far away, but I can see things close. All right? Again, let's just say the lens is too strong. Now, you're going to go to the ophthalmologist. They're going to say that, oh, you're nearsighted. We need to give you some corrective lenses. So what do we have to do? The lens has to correct for that over strong lens. So what does that mean? I need to reduce the strength of that lens. What does that mean? That means I don't want to use a convex lens. I need to do the exact opposite. So I need to use a concave lens, which is going to reduce that angle. My lens is too strong. I need to effectively weaken that lens. So that means I will put you with these concave lenses, which will then far away, they will account for the overstrength of the lens by modifying that and reducing that. So basically, they're making the signals coming in more divergent, and then the overstrong lens will then focus that, and then we will see things clearly. What about nearsightedness? Well, you still have functional accommodation, so then you can see near things with accommodation, right? So with those concave lenses, you can see things both far and near. Without them, you can only see things near. Okay, that's nearsightedness. What about farsightedness, which is this one here, all right, or hyperopia? What is that? That means that the lens strength is too weak or the eyeball is too short. Okay, let's just focus on a weak lens. A weak lens means that I can't see things far away, right? So basically, because I can't see things far away because the lens is too weak. But oh, wait a minute. If I can't see you because you're blurred back there, but with accommodation, I can see you back there. Okay? But wait a minute. What about when now we move up close? I've already accommodated. I can't accommodate on top of an accommodation. So I can't further increase the strength of that lens. So this means that I can only see things far away without corrective lenses, but I can't see things close up. All right. So what do I need to do? I need to increase the strength of my lens. So I'm going to use a convex lens. And that will be adjusted to me such that I can see things far away without accommodation. I can see things close up with accommodation because I don't engage it until I want to see things close up, right? So I do expect you to understand the differences in eyesight and these two, whether the difference between nearsighted, farsighted, and the corrective lenses that may be in there. What about stigmatism? I have astigmatism. I had an eye injury about 20 years ago, almost lost my right eye. It resulted in significant scarring of the sclera, that's the white part of your eyeball. What that resulted in is a warpage of that eyeball. So the eyeball is no longer this kind of round shape. It's kind of warped a little bit. What that results is that affects the ability to focus, which is uncorrectable with a lens. So I get in my right eye, I can never, it goes down a little bit every year. It's now around 80%. Okay. I can only see about 80%, and it's uncorrectable with a lens because of its astigmatism. It's not a lens problem, right? And the corrective lenses are only going to address this. They don't address, you can only do so much with astigmatism, all right? Okay. Getting close. So, how does it work? I mean, we're getting the light coming through the lens, and now it goes to the back of the lens with the retina. That means it has to be recognized by photoreceptors. Let's talk a little bit about how that works. Now, the light is coming through your lens. It goes to the back of your eyeball. The photoreceptors are not pointing out. They're pointing back to the back of the eyeball. So basically, that means that you do not see direct light rays. you see reflected light rays. So the light goes to the back of the eyeball, reflects back, then it's sensed by your photoreceptors. You have rods and cones, which I'll talk about in a moment. And basically what happens is, here are your rods and your cones. The light rays are bounced off the back. They're sensed here. It goes through these. Oh, look at here. Here we have these bi. Remember these? We talked about types of neurons. We talked about those with a single, two, bi, and then multiple. These are one example of bis. Then these ultimately are going to these ganglion cells, which ultimately feed into the optic nerve. And each eyeball has an optic nerve that then goes to the brain for signal processing. All right. So that's how we basically see. And that's all I have time to cover for this. All right. But I do want to spend some time, at least a minute or two, talking about rods and cones. These are the photoreceptors. And basically, let's talk about why do we need rods and cones? Why do you need two different kinds of photoreceptors? Because they're seeing different things. In the case of rods, these are in humans. We have around 100 million of them within each eye. They are designed for not seeing color, but seeing shades of gray. They basically are designed for low light perception. So they enable you to see in the dark to a certain extent, all right? And low light conditions anyways. So they have low acuity, and basically that's their function. Now let's go with cones. Cones are fairly unique. They're designed for high acuity, but they're also designed to sense color, all right? And that means day vision because you need high light quality. And they are low sensitivity. They don't see well in the dark, but they see well in the light. And they allow you to see color. Okay, so rods, cones. Now, have you ever heard that? I mean, I doubt very many of you have gone to a bullfight, but you see a matador, he's flashing around a big red cape. One side is black, the other side's red. You know, and you've heard, don't want to have red, you're supposed to attract the bull. Bullshit. Cows are colorblind. They can't see the red. They just see this idiot flapping this thing in front of them, and they go to charge it. Okay. So, as humans, you're primates. As primates, you are unique in the mammal kingdom in that we have a predominance of cones, which means we see color. Deers, lions, elephants, etc. They really have limited numbers of cones. They don't see a lot of color. Right? Think about your cat. Your cat has a lot of rods because it is a nocturnal hunter. Sees well in the dark, not particularly well in the day, but doesn't see color. You can see black, they say shades of gray. Okay, here's what I want you to think about. Think about those mammals, non-human. Think about cows. What is the color range of a cow? White to black, shades of red. What's the color range of a horse? Same. What's the color range for a goat? Reds and whites, blacks and whites, for the most part. Think about most of the mammals. Think about them. With the exception of primates, they're not colorful. Dull colors. That's because, what's the point? You can't see it, so, the only difference is now if we talk about primates, you've seen primates with a big red ass, they're in a certain estrous cycle, or you see they have these really significant blue markings on their muzzle. They have color. Look at all of you. Most of you got some kind of color on. All right. So that's the reason. So this is another strategy. Now I'm going to ask you a question. Having said that, which animal species see color? Anybody?
I presume birds have to because there are a lot of very colorful birds.
Yep,
out there.
birds. Next.
Butterflies.
Insects. Next. Pardon me? Yes, fish. Okay. We'll stop there. There's a few more, but think about it. Here's the easy way to guess. Do these animals have extensive cones? Are they colorful? Okay. Because what's the point in being colorful if no one can see you? I mean, if your fellow species can't see you. Okay. Now, in terms of insects or birds or fish, it can be used to attract a mate, or it can be used in many ways to as a protection against predators. Try to fool them. It's camouflage or something along those lines. Right? So not every animal species sees color. And of the mammal species, we and our fellow primates are pretty much it. That doesn't mean that a horse can't see kind of a reddish brown and things like that, but they don't see the range of color that you see. Okay? So let's spend a little bit time talking about color vision. All right. So color vision is a result of cones. You have three kinds of cones in your eyeball. All right. You have blue cones, green cones, and red cones. The blue cones allow you to see a pink sensitivity of around 420 nanometers. They have green cones at around 531 and red cones around 558. All right. So with these three cones, you can see a complete color spectrum. Right? Now, so basically, those means that when they're each sensitive to their own their own range of wavelength. All right. So what about color blindness? Now, when you get a large enough class, there will probably be someone that has some aspect of colorblind. It can be male or female, usually is more predominant in male versus female. And basically, what is it? I mean, I'm talking hereditary colorblind. That means that you don't have one or more different kinds of cones. All right. So basically, the most common is a red-green. So that means you like either one or the other of those cones. Okay? You don't either have red or you don't have green. All right. What does that mean? That means you can't distinguish between red and green. What it simply means, you kind of see this muddy kind of reddish, brownish colors, but you can't distinguish between that. Now, is it important? It can be. I remember many years ago, I had this was in the early 90s when, PowerPoint, I'm old enough to remember when all slides were blue and white because you took film and when you processed it, it was blue and then the text or whatever was white. That was it. There were no computers. You didn't have all that fancy crap you got today. Then in the early 90s, PowerPoint, and then, even better, you could translate that to print all kinds of colorful slides. Everybody went crazy trying to make colored slides because before everything was blue and white. All right? People were playing with all kinds of colors of background. What colors of background do you guys see today for the most part? White or black? There's a reason for it. Because depending on the colors, different colors like reds have issues in that they're not soft colors. They can excite you unduly. There's all kinds of reasons, but it also comes down to colorblind. I had a technician who was colorblind. And what I used to do is screen all of my slides through him. So can you imagine if you've got a red-green colorblind person and you have a red background and your text is green? They can't see anything. Right? Depending on the amount of contrast. So I used to pre-screen everything from him, and then you kind of get to the point where, well, wait a minute, what's going to happen when he's no longer with me? So I better figure out a very neutral color scheme. So everything is on a white background, and then you don't really even after all these decades, I kind of think about red and green. I always assume that if someone's colorblind, those are the chances. I try not to use those as distinguishing colors because somewhere, if I get a large enough audience, someone can't see it. Okay. All right. So that's color vision. So let's give you a test. Here's a test. How many of you see the number 78? If you do, talk to me after class. Because that's not 78. So you see there is the number 16. All right. Now, if you are red-green colorblind, that means you cannot distinguish between the red and the green. And that means that you won't see the green. You won't see the green, which is represented by balls of green. Okay. So these are actually functional tests for colorblind. This is real. All right. Okay, I have 16 after. I'm going to probably, let's do this. Let's do taste. Taste is fun. All right. If you've ever looked at a physiology textbook, especially an older physiology textbook, you will see a taste map. And that says that you can taste different things depending on where you put them on your tongue. You've probably all been taught that to a certain point. So basically, that means that you can taste a bitter, sour, salty, sweet, right? Now, this was originally proposed over 100 years ago, all right? And basically, I can still remember in physiology labs back a lot longer than you were around, we used to do that part of a lab group. We used to put stuff on there and see what you can sense, where you can sense salt, where you can see sweet, etc., and so on, all right? It works up to a point. What was ignored for very long was an addition to bitter, sweet, salt, sweet is umami. Okay. This was proposed really quickly in the early 1900s. And however, it wasn't, and that translates to meaty taste. This does not actually make it into the taste map into the 1970s. Or 1980s, excuse me. Okay. So I kind of like to put that in there. So here's the modern map here. Again, just like in those referred pain, your taste buds, generally speaking, but you have the ability to sense other things depending where you're at. So in other words, even though most of the sweet is here, if I put it back here, I can sense it too. Okay. Or salty, I can sense it here as well. If you put the tip of your tongue on a piece of salt, you can taste that. Okay, so these are just general areas, but there's a lot of overlap. All right. So basically, this is the way your tongue basically works. Bless you. Okay. Okay. It's 20 after, so I'm done. Okay, so we're going to stop here. I don't have time to go anymore. Bless you. So we will stop the nervous system at the taste. That's it. Everything else, we're not going to get to the brain or anything like that. So that will not be on your exam for next Thursday. All right. Thank everybody, and I'll see you Thursday. If I'm not mistaken, you might have a quiz on Thursday as well. That'll help you for next week. Thank you.